Okay, so welcome. We want to predict from the input of an amino acid sequence which residue in the protein is binding to other proteins. We do not know the binding partner, we do not know the structure, we just take sequence and we want to predict. How can we choose the input features to learn this? My first question before we get into this, how do you code the input? Not sure whether we discussed this already on Tuesday. So I want to predict which region in the protein binds. So what do you put as input into the machine learning device? If somebody feels cold, maybe you want to close the window? Okay, I won't. There seems to be somebody feeling cold in the room. I'm not the one because I move. How do you do this? So far we have discussed, for instance, the prediction of subcellular organization, where you use the overall amino acid composition. You cannot do that here, right? That's clear to everybody. So how can you do it? It's fairly simple. It's just
residue number of residues.
So the number of residues, so you have a sole input?
Position.
The position. What do you put in then? So you put in what do you mean by position?
So if you understand like the question, we need the position of where we have some connection binding interaction.
So let's just look at my protein and say here are binding sites. So this is my protein. So what is put into my... This one goes more into detail what exactly we use as an input feature. My question here is more generic. What's the input? It's something derived from sequence, but so we cannot use the entire sequence. So Gary said we use this, and that. Is that what you meant?
Yeah.
So nothing else. If you do that,
not nothing, but at least that.
well, the problem is if you could do that, right? So you could train on these segments that bind. What's the problem with that? Yeah?
Couldn't there be another binding site?
That is also true. Yes, that certainly is true. Yes, in fact, that is partially already my answer. The thing, there could be a binding site, and ultimately you're developing a device in order to find that dotted binding site, right? And if we did that, then we couldn't find it maybe, but more generically, if we train on these three regions, what is the problem?
Maybe they're not on the surface.
Yes, but that could be true what you say. But we are training explicitly on binding sites. So they may not be on the surface, but if I want to predict binding, so protein-protein interaction, so all of these, so the black ones here are PPI sites. And them being on the surface or not, whatever they are, I want to learn that. So that may be, but this is not the major problem, yeah?
Maybe all the sites that we know are of a certain type and all the ones that we don't know are of another type, and so
maybe we don't have generic information. True. That is not what I mean. There's something much more fundamental in here. Yeah?
Well, basically you need to have the machine do a decision. So you have to say,
yes,
this is and this is not, otherwise...
So I clearly need these sites. Because if I run my device tomorrow, and if all I've ever seen is these black sites, then all the system can learn is I say whenever input comes, binding. Right? This means that if I ultimately apply the device and I sort of shift this through the protein sequence, it will always say binding. We need a counterexample. We need the negatives. So we need the non-black regions here. And yes, you're right. So we would, in this particular case, train on something that in fact is a binding site. We don't know, we make a mistake. True. That is, in some cases, maybe it's not on the surface and I look at surface composition, so I'm missing some things. That is all true. But the most important thing really is I need black and white. I need binding and non-binding. I need the classification. I need different things. And then I will ultimately move my window through, right? But the next question is, when I imagine a window of, say, 5 consecutive residues, so I'm going to move 5 consecutive residues, and I don't know the 3-dimensional structure, so I cannot look at 3-dimensional patches. I cannot actually say whether it's on the surface or not. All I can use is sequence for 5 or 5 or 9 or something consecutive residues. But now you get the task. And you're sitting somewhere in your office or you're sitting at home with your computer. How do you choose input features? Your first algorithm will always be Google. So you will look up at Google, what did others do? So there are 2 issues with this. If you only do what others do, then this is not going to give you a bachelor thesis. Or you're in the level of master thesis. So clearly not going to give you a master thesis. You have to do something new. So you at least have to do some random permutation on the Google find or something. But what else could you do? So here comes the point of networking. At least ask everybody around in your group. Talk to people. And so ask people not necessarily where do I find the water to cook tea. If you can do that, but if you do that every day, then they may not like you so much for it. But if you tap into their scientific intuition, that is something we're all proud of having. Over the time you evolve scientific intuition. If you talk to people in the lab and you get them to the point where they feel that they know something, this is good for you and it's good for them. So don't stop this discussion. Keep pushing on levels like that. Not on levels where's the Perl compiler or Python compiler. Those things are not good. But things that sort of are more into somebody. Anyway, this will give you one idea. Now ultimately, when we get into this input for information story, we will get a classification device. In this particular case here, I show a window of 13, not 5, 13 consecutive residues. And the 13 consecutive residues are such that the P in this example here is at the center of this window of 13, okay? And what the device has to learn is what is the state in terms of protein-protein binding or not of the central residue. So the central residue in this particular case here, indicated by the brackets, is in the P state, in the binding state, okay? N is the non-binding state. But again, this device, this particular window here, has to learn that this unit should go on, okay? Now, each one of these blurbs here is 21 points, 20 for the minimal way of using sequence. So we get back to this issue, what can we use as input sequence, input information, but minimally you take the sequence. And one way of doing that is you use 20 units here in a sparse coding. One of these 20, in this particular case the P here, is 1. All others are 0, because of the proline at that position, right? And so in this one here, that unit would be on and all others. Why do I need 21 units? See, is anybody in the room who has not done last semester? I believe so. Any idea why we would need 21 units, not 20? The 3 of you.
For unknown amino acids?
That is a good idea. That's a very good idea. But there isn't even, in fact, people have done that and have used that particular unit for that purpose, and there's a reason for that. Yes, but there's another reason, or a more dominant reason. Let me call it that way. Okay, so this device is going to predict what is the state of the central residue in this window, right? In this particular case, it's in a P state, okay? So in this way I show it, there are 6 residues before it. Yes? Does that give it away for the 3 of you?
Maybe if it's at the beginning already.
Exactly. So what if I want to predict if the first residue in the protein should be predicting? I have 2 options. One is I can say, okay, for the first 6 I will not make a prediction. Then I only need 20. Okay? Or I introduce a new amino acid, which may be the unknown, but most importantly will be a spacer. So will be before. So will be all the ones that are not amino acids. And in fact, when you use unknown, people used a 22nd unit. That's typically what they did. And unfortunately, that doesn't work. Or at least in my hand, it didn't work. And my rationalization for that was that I don't have enough of these samples. This is just one unit, so you build up an input vector of 13 consecutive, and in these 13 you have 13 times the new unit here, that in only one of 20,000 samples is used. And for me, that was the explanation why it didn't work. I tried that. But the spacer unit is crucial, because this essentially is teach the system you are now with a window at the beginning of the protein or at the end of the protein, right? Okay, clear for the three of you? It's relevant for the three of you because for the people who have done it last semester, there was the same concept, and in fact, there was the same graph. Okay, that's the simplest, and in the simplest solution, we would have one output unit. And how could we then classify into P and non-P? So the final step is a sigmoid function that goes between 1 and 0. So this is 1 and this is 0 here. So this value times that value plus this times that is summed up. And then there's the sigmoid function applied. So very highly positive values are switched to 1, and very highly positive and negative values are switched to 0. So if I had only one output unit, how would I distinguish between protein interaction and not?
Maybe a cutoff and everything above the cutoff is...
Exactly. So you would typically, the default way, the training is completely symmetric. So the default way is 0.5, so half. If it's higher than 0.5, it is classified as protein binding. So why could it be an advantage to have 2 units? That would essentially do the trick. So why could there be an advantage in having 2? Where then what you of course will do, if you had to, just let me continue on this statement, you would not say that is higher than 0.5, you would say if the p-value is higher, the value of the p-value, no matter how high it is, if it is higher than the non-p value, we would say p. If it is lower, you would say non-p, non-binding. Okay? So what could be the advantage of having 2 units? So there may be some correlations between particular input vectors that give a low signal for both. And they are not giving a low signal in the sense that they are undecided. There may simultaneously be a signal that goes into the direction of non-binding, and so it could be that this unit is close to 1 and that this unit is close to 1. The average between the two is still in the center, but in this particular case you reach this average not by an undecided decision, but by a flipped coin decision. And that is a different situation that may contain important information. And ultimately the way you learn this important information is because you simply have an additional layer here. You have an additional set of connections that can learn this. So they can learn correlations. It's particularly helpful if you have more than 2. So if you have 3 states classification or higher order state. But even for 2, this in our group, almost always when we tried, was an advantage to using 2 units over 1. And ultimately, also going back to this decision that works better. So in this particular decision, what I show here is in both of these, so this is input vector 1, this is input vector 2. In both of those cases, the prediction is this is protein-protein interaction because that top unit is the highest. But in this particular case, the decision is much stronger than in this case. The strength of the prediction translates to a reliability index. So you can essentially measure on the level of the prediction how reliable the prediction will be. And we have to establish that this correlates with accuracy. That I will show you. And that typically works again better if you have 2 units.
Question. So So what would be the stronger prediction if you have 0.12 for the first and 0.10 for the second, and the other one has 0.8, or let's say 4.8 for the first and 4.2 for the second?
So that ultimately we would have to analyze those two situations differently and essentially ask the question, is there additional information? Are there cases, what are the cases where both units are very strong? What can I learn from those? And in those particular cases, typically what we have used this is to influence the training. So we have kicked things out from training. We sort of reduced the training data by assuming that this is sort of ambivalent information. Now, in that particular case, then the answer to your question is, it depends on your definition. If you define as the difference between the two, then they are equal. Right? The reliability between the two is equal. But there's additional information in it, and that you have to use in a different way, not for the reliability. What the additional layer allows me is to have typically a better mapping of the reliability, meaning you have a better function. So with increasing reliability, you have increasing accuracy. And that, I'll show you that curve in a moment, and that typically is helped if you have two units. Back to the question of the input. What could be an important input? We want to predict whether or not the central residue in this window here is in a protein-protein interaction. Here I only show sequences input. What else could you put in? What would you put in?
You can put distance from beginning or end of the protein.
That in fact is an important part. Yes, we do see somehow the distance for cases where we're touching the end, so where we have these spacer units on, the system can somehow learn that I'm close to the end. But it cannot learn, in a window of 13, it cannot learn whether it is 15 residues from the beginning or 50. So this as additional unit is something that helps, yes. But this is what I would call global information. What could we still put into the local information here? Something that is sort of relevant for that window. Can you say a few things? Yes?
Charge, hydrophobicity.
So we could essentially take sequence and put some, I'm not entirely sure how to phrase this, functions upon it where we map it to biophysical features. Our intuitive understanding of the biophysics. Charge is one example for that. Hydrophobicity is another. Yes. Could even compile an hydrophobic moment. In this particular case, that does indeed help. Charge in this particular case also helps. Hydrophobicity also helps. So that would then increase. So one way of doing it, in fact this was the way we initially did it, instead of 20, then if you use hydrophobicity and charge, you would have 20, instead of 21 you would have 23. And then you could do that differently. You could, in some cases we did only these additional features for the central one, and some we did it for the entire window, depending on how many samples you have. But what else? Other information that you could imagine. So it cannot be 3D structure because we don't have that. Yeah?
Predicted solvent accessibility.
That's a very important one. Something else.
In coarse grain simulation, so for each amino acid you can assign like equivalent mass or radius or something like that. Can we extract?
Size. Yes, I do not remember whether that helped us. So charge and hydrophobicity did. I do know that the size helped for secondary structure prediction. Yeah.
What about flexibility?
Yes, that helped. So I briefly introduced the B-value prediction, right? When we talked about where to put the threshold, and that method is also used as an input here, and it did help. Yeah.
Conservation.
And that's, now we're getting into something else. So you would run an alignment against the database and would ask, is this particular P here conserved or not? Is that what you would put in?
You can try it. I don't know.
Yes, that does work. And that has helped. But there's something bigger. Or no. Well, so let's just wrap it up. Secondary structure also helps. is also an important feature. But on the level of conservation, so one should think about conservation. Can you? Well, yes, conservation helps. What else? Conservation is one value, yeah?
I mean, it's sort of related to conservation, but not
Yes.
exactly. It's
Yes.
to see if this amino acid is substituted sometimes by something that is similar. So if
Ooh,
you have
that is a-
line and these are lines,
I do not know how you come to that idea.
But there's metrics for that, isn't it?
Yes, but nobody has ever used that as input.
Okay.
We just submitted a manuscript. We have done that for the first time, and it actually. So I was proud of that idea. It worked actually like a charm. But we have not used that before. Great idea. Works amazingly well. So we haven't done that here. So we have just Maria Schelling. She does the exercise with you guys, right? She just submitted this, and it works better than the conservation. It works better than anything else that she tried for some other problem. But essentially, predicting binding sites. So DNA and enzymatic activity binding sites. But I mean something along more directly related to conservation, but sort of conservation is a part of it. Okay, the alignment information, the entire alignment. Yes, conservation is just one value of the, but ultimately we have in each of these blobs here, we have 20 amino acids, and one is 1 and the others are 19. We should replace that by what we see in the alignment. So rather than replacing here in this particular case, it would be the same, since all, so that is the running protein sequence. Those are the 4 relatives aligned to it. You see that the D is always D, the P is always P, but here, at least already in the E, you see that the search, the query sequence for which I have the annotation where the binding sites are, has an E. All the relatives, or most of the relatives, or 60% of the relatives have D. Only 40% of the, only one relative, only 40% of the sequences have an E. So the E becomes the minority. So E and D are both observed at that position, but the sequence position, or the sequence, the residue that's actually observed in the annotation is the minority in the alignment. And I may be right or wrong, but so we would replace then the 21,119,0 vector by simply this count. Or by the psi-blast estimate of importance. And then the question would become, would it still help? So implicitly, you have the conservation in here, right? And if I now asked you, okay, I put in this entire alignment information, my system learns that, and by the way, that is the most important single feature here. Now I ask, should I add another unit? So 13 times 1 at least. So conservation, 13 conservation rates. So it's a window of 13 consecutive for each one of these blurbs. I'm going to, on top, so I have the alignment information in there. And am I going to use on top another unit, the 22nd, with conservation rate? Yes or no? Is it going to help you or not? One says no. Who joins in the chorus of no? Who joins in the chorus of yes? Yes?
I think it's sort of redundant. It's nice as fast.
That's why I assume that's why you say no. But ultimately, well, Lucas said no, Franziska. But the important point here is we do not know. The only way is try out. Again, for almost every single case that we looked at, it did help. And the reason for that being, it is sort of redundant, but it's actually not. Because what you do not see in here is what's the overall variety of these sequences. So if you now had, and you already begin to see that because they are very similar here, right? There's a lot of similar residues at all positions for this example that I chose. And you do not see whether there's 100% in one particular D because there's no diversity, so all the sequences that you used are the same. Or is it because, in fact, that is a completely conserved residue? And that would be reflected by a conservation rate, which looks at the overall percentage sequence identity and weights these scores that they see. Right? So it's sort of redundant, but actually not, because this overall global information is not available on the local profile. So PsyBlast tries to somehow weight against the Blossom matrix, but still does not put the overall weight into account. So this information is still not in there. So it may help. Okay. Now, by the way, if you don't have an alignment, what would you do here? Would you go back to 19 and 1, 1 and 19 zeros, or what would you do? Say you had some, it's increasingly not the case anymore, but say you had some protein, so say I already told you that this is the most important single feature to do the prediction task, right, the alignment. Say now you're going to predict for a protein for which you don't have alignments. So you search the entire UniProt, you don't find any homolog. So then ultimately what you're going to put in is what? A single sequence, but again back to 19 and 1, 19 zeros and 1 1.
Maybe you just use a standard matrix like
the Blossom, and that again very often helps. It's a very simple hack that typically helps, and this is what you have to assess. You have to see whether for your problem that's true, but there are still cases, even for human, there are still families where this is the case, and that may or may not be relevant, in particular when you use the sequence variation effect prediction. Okay, so we named them all. The only thing that we forgot, but this is like secondary structures, membrane region, disorder we forgot. The predicted aspects of function is mainly flexibility, and mainly flexibility at this point. Okay, now are we there yet then? Well, then we have to do cross-validation, right? We have to sort of hide some of our data under the table. And then essentially we get into a loop. What is this? This is one way of making the talk longer. I'm sorry for that. So when you do the cross-validation, so first of all, in order to do a proper cross-validation, you have to always begin by preparing your data set such so that's your experimental information, and you take some subset that is such that no pair in the subset has more similarity than some threshold. So it's non-redundant, unbiased, or whatever you call that. Now, my question is, when I do the cross-validation, can I just randomly split this set E and take whatever the number is, for the sake of the argument, 2/3 for training, 1/6 for cross-training, 1/6 for testing, and publish the 1/6 for testing. Is that a valid algorithm? It's a correct algorithm, but is it okay? Is it a proper behavior, or whatever you call that? If you had me as a reviewer, would you get through? Yes?
Well, if the distribution between the classes of binds or does not bind is very different, then you have to make sure that so that the distribution is sort of okay in all of these cases.
So let's just say that we get back to that point. I'm not going to answer whether that's true or not. Let's get back to that point. Something else. Some other concerns, some other issues.
Are we talking about splitting capital E or
No.
small e?
So again, we're talking about capital E is reduced to small e. Whatever is not in small e is thrown away for the time being. So we are splitting small e. That's a good point, because if not, then you have to absolutely make sure that there's no overlap between training and testing. But in this particular case, by the definition of small e, there is no overlap. Otherwise, we change the small e. Guido. And I'm sorry, I still don't know your names. But maybe if you stay with me another 2 or 3 months, maybe then. So, why, let's turn that around. What's wrong in it? By my way of asking the question, it's clear that something is wrong in it. But what? And we will get back to the balance story. So again, for those of you who didn't understand that point of Franziska, what by balancing she means, say in the case of my protein binding sites, say 20% of my residues are binding, so 80% are non-binding. So what Franziska means is that I have to make sure that on average for the training set and for the testing set, these numbers are roughly the same. And again, I'll get back to that later. I just want to explain that point. That's not what I'm concerned about. That's not why this algorithm is entirely wrong. This algorithm is entirely wrong is because you have to rotate through it. It could be that the way you choose it, by happenstance, the test set that you chose, this 1/6, is one that is a lucky case. You have to rotate. You can't choose at random the way you rotate, but you have to rotate such that every single protein in your data set has been used for testing exactly once. You cannot oversample, because again, there will, for whatever we have looked at in our work, there was always some distribution. So where here's the distribution, and here's the performance, there's always some, it may not be quite a Gaussian, it may look like slightly different, but there are always some proteins for which performance is better than others. And by randomly selecting, you're sort of picking random points in the distribution. Okay, the distribution that you see today, the proteins that you have today, may not completely reflect tomorrow. But still, the best thing in some sense is that we can begin to sort of assume it does. Okay, so that means you have to have many more rotations. In fact, you have to do this 6 times. Okay, exactly 6 times. Not roughly, exactly 6 times. Now, the balancing story. So there are 2 different words for balancing. So let me begin with a word for balancing that I understood in what you said, which is in fact that I should have a similar distribution in all 3 data sets. The more similar the distributions in all 3 data sets, the more likely my performance is high. So the more my training set is like my testing set, the higher my performance. The more different it is, the lower the performance. Meaning that if I choose the split such that this is the case, then I choose a parameter that helps me to get a higher value in testing. Is that good? You remember my strong statement? Never use the test set for any decision. The split now is a decision that you make. And you in fact do this in order to optimize the outcome. Do not optimize the outcome on your test set. Okay? Even when you do that on your cross-training set, even that I still would feel tends to make you overestimate performance. You may call it more fair. It is clearly helping your prediction method because you create a situation that is most similar. I wouldn't do it. But clearly don't do it for the test set. Right? Clearly, maybe you do it for the cross-training, but clearly not for the test set. Because then you essentially use a parameter that you know through your labels that you shouldn't use. Okay, so let's get back into the performance story. What I'm going to show you now is a curve where I go through various levels of accuracy versus coverage, precision versus recall by using different thresholds. You're going to see on the left of the next side of the next curve, you will have a higher threshold. On the right, you have a lower threshold. And this is essentially the spectrum you get: coverage, so correctly predicted by observed, versus accuracy, correctly predicted by predicted. And this is the curve. That's the entire curve. So there's nothing else to be seen. On the next slide is the same curve, it's just stretched. So the axis now, as you observe here, is 0.03%. So I get a tiny fraction of the data, and that is the level of accuracy I get for the tiny fraction of the data. Okay? And that is sequence alone. The background here is some random model where I simply take the predictions I have and I randomize the labels. So I take, for every single protein, I take the same number of predicted residues predicted to be binding sites, and I just switch the labels randomly. That's the way the random is created here. And you clearly see that there's a realm for very, very, very few predictions. You're clearly away from random. Okay? And what does it mean? So it means that, in fact, at that level here, we get at least one prediction in the protein. In every single protein, we get at least one prediction in every protein that we know binds other proteins. On that level, we have at least one residue predicted. Okay? Which is, well, protein interaction typically has 20 to 30, 40, 50 residues, so 1 in 40 is a small number, but it's a start. Okay? And for something that looks in some sense so dismal, in particular if you look at the entire plot, and it's still not the entire, 70% only here, this is something, right? And then when we look at the 3D structure, we see that it's not quite completely right, but it's at least close to this actual binding site. In fact, this labeled binding site, and it fits. So this one prediction is by all means correct. Now, in order to do this, I need to set up all kinds of things. Okay, so the simplest way to improve over what I had before is some averaging process. The averaging process looks at, rather than looking at the neural network output, it says, okay, I now look at a window of 9 consecutive residues. And since I know for real proteins, in the 9 consecutive, the blue is what counts here, at least 6 in a window of 9 consecutive are binding. I simply run my predictions and I say I average over windows of 9. Right? And that already is improving. But then I put the alignment information into the system, and then that's what I get. So this is sequence only. This is using alignments and secondary structure prediction, solvent accessibility prediction, hydrophobicity charge, prof B value, so the flexibility prediction, and again, this is random. Now you see we're clearly away from random, right? Now the story becomes the topic of hotspots. So let me go into the hotspot story. Are you guys familiar with this term hotspots of protein-protein interactions? So one way of saying it is when you have the interaction between two proteins, there are some residues in there that are more essential than others. How would you measure that? Well, one way of measuring it, you would simply say, what's the contribution of a particular residue to the free energy of binding? And it could be that there's one residue contributing that much, that the second one is contributing that much, and you have some curve like that, right? So you have some residues that contribute much, much more than others. And these you could call hotspots. There has been a long debate about whether it's reasonable to call them hotspots. For drug design, that is essential, because drugs are typically exactly targeting these residues. They tend to want to interfere with other binding, and the best place to go is the one where you can sort of knock out with one connection most, right? So if knocking out this residue carries half of all the free energy of binding, then that's exactly what you're going to develop the drug for. That is why this is an important issue. Are there hotspots or not? Now, the people who attack this idea of hotspots, they say what you have here is a function. In this function, just like in the example that I had before in the B value, if I asked you where do you choose a threshold if you want to learn a binary system, you cannot say. Any threshold you put in here Well, typically, you know, for medical testing or in chemistry, you have an IC50 score. You have something where sort of half of but half is a completely artificial number. Whether it's half, is 30%, 60%. In the curve here, there's no difference. Yes, we can compile an IC50 because in chemistry we have learned to think in terms of this 50% half-life or things like that. But in terms of, is there a distinction between the ones on that side and that, other than that is more than that? It doesn't make sense, right? This is why the people who argue against the existence of hotspots simply say, well, that's a curve and there's nothing, the numbers are different left and right of this threshold, wherever you put the threshold. Left they are higher, right they are smaller, but there's nothing systematically different. There's no feature that you can look at and say, this is going to be a hotspot. It's defined by a particular interaction. Okay, now, in our prediction method, we have no idea about hotspots. We simply predict, we take bindings between two proteins. We trained all the ones where we have less than 6.5 angstrom between the closest atom, we call binding sites. And the other ones we call not binding sites. That's all we train. We're not labeling hotspots, okay? But the question is, maybe the ones, so you see there are a few residues that are predicted very high, at very high accuracy. Very few of those, wherever you put this, but somewhere here, maybe those are the hotspots. And the person who did that, Janai Opran, was sitting next to somebody who did a large alanine scan experiment, again on GP120 binding CD4, because it was in the lab of Wayne Hendrickson. And experimentally, they found the red ones to be hotspots. And Janai, just looking at the very top predictions by looking at the strongest predicted, so not looking at which ones are right, but simply the ones that are predicted the strongest, found the purple. The structure here is just an overlay showing it's not used, it's just to visualize it. Visualizing on the structure, you see that sometimes here we miss one in between, we over-predict one in between, sometimes we miss one in between, here this one is missed, and so forth and so on. But given that all we needed in order to do this prediction was sequence, this is a remarkable result. Is that enough to publish? Well, actually, it was enough for Janai to give his thesis. But is it enough to publish? No, you somehow need to get at a data set where you have more than one. So in his particular case, so there was only one experiment so there was one friend he had, that was the one who sat in the office next door, and he asked that one, and this is the only case he ever tried. So it's defendable, but it's still a case that is difficult to make in journals. Because people have to trust you. You tried one, but how would the other one look? Maybe you were just a lucky kid, right? So you need to find more. And there is a database of alanine scanning variants, and that is what he used for the next step. The numbers are overstatements, we believe today. We have redone this method and it doesn't work as well with larger data sets today. But in principle, the statement remains true. So this is now the same neural network applied to predicting hotspots for proteins that had never been used for training. So the concept of hotspot had not been used, and these proteins had not been used for training. So it's completely cross-validated because of a new data set and a new task. And then again, I can ask, do I recover the experimental results? And you see that at least for some of those here, in those cases we believe for 10% of all the proteins, we get all the hotspots right. At that threshold, again, it's driven by the reliability of our prediction. We don't make a mistake in getting the hotspots. We don't believe now that we get 10%. We have to redo this plot here because the underlying other plot, this one here is no longer. We are not reaching this level. We tend to be more here. So the more recent version of this one here is below. Using more features, more data, and everything, more things got more complicated. But anyway, so the real aspect here is we have a method that learns to predict protein interaction sites and can actually be applied to predict hotspots, this implies that hotspots, whether they can be predicted. If they can be predicted, then there is a difference between the ones on the left and the right, no matter where you put the threshold. So hotspots exist because you can predict them. And you can predict them by a device that has absolutely not been overfit because it never used any of the data. So this is an example where you use machine learning to settle a biological or biophysical question. So it's not, again, this device is used by people to predict hotspots, but in this application, in this view, I'm using it to say, well, it makes a decision for a long-standing debate. You had a question, Guido?
Why is the random model at about 21%?
So we simply looked at the average that you get if you predict 21%. So 21% was the one that we saw. So we flipped the coin at the proportion at which it was in the database, and then we had a flat number. So here we did not randomize the prediction. Okay, so again, the most important thing was the evolutionary information. Explicitly using the conserved residues did help. Importantly, predicted 1D structure, surface, secondary structure, low complexity, sticky regions, so disorder, and filtering isolated predictions. Here again is how you improve from just using hydrophobic moment, sequence only, evolution only, to using all the information. Now you can use this method and in fact try to see whether you can find alternative binders by proteins that have similar predicted binding sites. So you look at two proteins and you simply say, well, those are my binding sites. If I have 2 times predicted the same binding sites, maybe these proteins bind similarly, and maybe I can use that to look at adverse drug reactions. And that is something where we have some preliminary results, and that essentially is it. But let's talk about a different issue, the issue of hubs. On the network level distribution, so on a network level distribution where I'm asking how many protein interaction partners does a particular protein have? And the first question that I have, will all proteins have a similar number of interaction partners? So phrased in terms of this graph here, if I plotted the number of protein interactions of one protein, so how many interactions does one protein have, and here's the distribution, which of these 3 curves do you expect? So do you expect that essentially every protein has the same number of interaction partners? Do you expect that there is some sort of optimum, a Gaussian distribution? Or do you expect something like the green line? Some proteins have few interactions and some have a lot. And what I'm asking here is not what do we know is today in the database. The database we all know, in particular for protein interactions, is biased. What do you believe will be in the database tomorrow? What do you expect the true answer to be? Yeah?
I would expect it to look like this graph over there.
Yes, so you're 2 steps ahead of me. Ultimately, I'm not entirely sure. You have seen this, you have most likely read the papers. Let me slow down and ask you, what would you expect if you plotted the size of villages? Here's one solution. From 91 US metropolitan areas. You can use any kind of data. That's a log-log plot, so it's exactly what you say is the 1 over x is the zip, the so-called zip flow. And Zipf, after Mr. Zipf, who actually was an American who studied in Germany in the 30s and counted word occurrences and tried to simply see whether by counting the number of letters he sees in a written document he can distinguish between German and English. But just counting how often you see an E in a particular document. And you can, because that again follows the Zipf law. Just like settlements, just like size of train stations, just like any system that you put into your computer and you simulate yourself, where all you need is that the evolution or the growth is under some constraint. So there's competition, is a way of saying that. As long as you have competition for resources, so to speak, you will always get Zipf. Okay? And that in fact is exactly what you will have for protein-protein interactions. Now, if you picked at random a subset of Zipf, so if tomorrow's protein-protein interaction distribution were to be Zipf, and you only measure 10% of the proteins, what will you get as a distribution? Actually, it's the same answer for a Gaussian. It randomly chose half of your. Not if you explicitly you only choose this side. So if you cut out one side, that's an unfair game. Then the answer clearly is not. You get something very different. Yes.
But the most data points are in the latter part, so it will look more like a line.
Depends on how you decide the most data points. So. No, but so randomly you will not select by proteins. Randomly you will select by interactions. And then the statement is no longer true.
Then it will
That's the crucial difference.
more like a line.
No, actually the random subset of a zip is a zip. The random subset of a Gaussian is a Gaussian. Half a Gaussian is a Gaussian. Half a zip is a zip. So as long as you're not deliberately sort of cutting either side of the extremes or something like that, as long as you're not specifically choosing hubs. So when cancer was first analyzed, it was P53. P53 is one of these cancer hubs that comes always because it's such an important protein for all kinds of pathways, not for cancer, for anything. It's an important transcription factor. And so if there is a bias of experiments for some, but if you do high throughput and you're relatively unbiased, then you're actually going to get a zip. You had a question on an issue?
It's sorry.
So, okay, and that is work I'm going to show you now. Work from Avner Schlesinger and Jana Ofran. So now we have a method that predicts binding sites. Okay, what could you do with it? I'm missing a slide in between here. But I can as well show Janai and Avner. Both are a little bit older now. Bailan in Boston University. So he's Professor Bailan at Boston University. And he's a professor at Mount Sinai in New York. Anyway. What could we use in the context of networks? What could we use the prediction of binding sites for? Any idea? Any suggestion? Nobody? I'm over your head. So ultimately, what I do with my binding site predictions, I look at a particular protein and I predict a micro level of biochemical function, so to speak, right? And there's another level where I look at networks. I predict, starting from a sequence, this protein has 5 binding site residues. I don't know anything about these partners, right? But maybe if I have a lot of binding sites, I get a lot of interaction partners. Okay? Maybe I can somehow find a correlation. And if I could find a correlation, maybe I could distinguish between what side of the zip your protein is on, whether it's one of those hubs or not. So what's the immediate problem that you see with that? There's immediately some hole in my argument, something where it will absolutely go wrong. Can you think about that?
We can have several proteins interacting on one side.
Yes, exactly. You take this protein that has one binding site, binds this protein, binds that protein, binds that protein. It has one binding site binding many different proteins. That is absolutely not going to give you more binding sites because it's the same number, and still this one goes up, right? So it will absolutely fail there. And in fact, this is Marc Vidal is one of these people. Marc Vidal is the one person who pushed, I believe, yeast to hybrid more than anybody else in terms of becoming a reliable source and doing high throughput screens. And Marc Vidal also introduced this term of date and party hub. Date hubs essentially are exactly those. Those are the idea when you go to a date, your face is looking at the person you're dating, and the same attention part of you is focusing the attention on that dated person, right? While when you're at a party, you simultaneously speak to many people around you. So essentially you have several sides of access. You talk left and right, you talk behind you, and that is the party hub. It's what we do at parties, right? Some people do it better than others. Now, in this context, then, where we would see this correlation that I talked about here is for party hops, right? Not for date hops. So let's see whether I can see a difference between date and party hops. And the answer is here. So essentially more binding sites predicted. That's the party hop. This is non-hop. And that's date hop. When you look at the way they overlay, so the gray and the purple are really on the same. If I don't look at 3D, they're really on the same spot. This and that, they are really identical here. That's why you hardly can see the gray behind it. And the other one is really different. Okay, this is not like the linear correlation. This is not as simple as I may have wished it to be, but it clearly establishes a link from the micro to the macro level. You do see some aspects of a network, although you don't see networks. So you're really on the molecular level, right? Any idea how we could sort of attack date hubs? Can you think about some feature that you may expect for a date hub? Some of you may have heard that last semester, but I can't remember whether I discussed it last semester. Sometimes in the first lecture I discuss it, sometimes not. So I can't remember. No? So one issue is if we have disordered region. And sometimes disordered regions are exactly the ones that allow you to bind with the same binding site to different molecules, because it's more flexible, because it can adapt itself to different environments. And this is in fact one particular type of disorder, the loopiness. So these are essentially long loops. Loops meaning not regular secondary structure, no helix, no strand. And you see this is now completely overrepresented in the date hub. Not in the party hub, and here, in fact, the non-hubs are even less. But so somehow you can spot that, and for particular proteins here, you can, in one case, really show a date hub that you can predict through this. In one case, you can predict a party hub.